Hello, my name is Andrea Radici. I'm a PhD student at the ERA Institute in Avignon, and today I'm going to show you the results of a work I conducted with the supervision of Davide Martinetti and Daniele Bevacqua at the beginning of my PhD experience. The title of today's presentation is Surveillance of Airborne Plant Disease Dissemination at Continental Scale using Air Mass Trajectory Analysis and Network Theory, and takes as case of study Puccinia graminis, which is a mushroom and airborne pathogen causing stem rust of wheat, a disease considered to be virtually eradicated in Europe in the 70s. The symptoms of this disease appear like red rust on the stem of wheat, hence the name, and its spores can travel several hundreds of kilometers transported by wind. In recent years, interest in Europe was raised by the detection of a new race in Germany in early summer, causing several losses in wheat production, and later transported by wind, was observed in Denmark, Sweden and UK. So the objectives of our study were to model the connectivity of air masses over Europe and the Mediterranean basin, to explore spatio-temporal patterns of connectivity, and finally use this information to identify regions associated with high risk of spore dissemination at continental scale. We modeled the air masses trajectories by means of backward Lagrangian simulations with reversed wind fields. First, we chose an air mass in a specific site in a moment in which rain was occurring to simulate wet deposition of spores. Later, we simulated its backward trajectory in the previous 72 hours using iceplate software. In principle, whatever site intersected by this trajectory can disseminate its spores to the rival cell. We imposed a set of filtering criteria to identify those sites which could effectively lead to a dissemination event. First, unsuitable timings of release were discarded, since we know that Puccinia spores are released during daytime mainly. Also, sites whose land cover was not compatible with spore release were removed. Finally, sites associated with too high values of cumulative rain or solar radiation with respect to spore viability were discarded. At the end of this process, we selected all the sites that could have realistically disseminated spores to the rival cell and summarized this information with an adjacency matrix. We repeated this experiment for each of the about 4,000 cells of the domain. Each one is a 0.5 times 0.5 degrees site covered with wheat for at least 1% of its surface. For each of these cells, 72 hours backward simulations were run using iceplate software. The simulation was repeated every 3 hours from the 1st of January 2013 to the 31st of December 2018, obtaining almost 70 million of trajectories summarized in 18,000 matrices. To deal with such an amount of information and extract the prevailing characteristics of the air mass through connectivity, we performed a two-level aggregation. The first is temporal. We aggregated the thousands of matrices on a weekly basis so that to get only 52 matrices. At the same time, in parallel, we aggregated the cells according to a spatial criteria into the so-called NUTS-1 regions, where NUTS is the geographical statistical classification by the European Commission, so that to reduce the number of cells from more than 3,000 up to 137. These spatially and temporally aggregated matrices were thoroughly compared using a clustering algorithm based on the cut distance, which allowed to discover a two-seasonal pattern that roughly corresponds to winter and summer seasons. This discovery agrees with the known wind patterns across Europe from atmospheric science. Here we show a representation of a reduced version of the clustered matrices, the so-called backbone. Winter on the left and summer on the right. Even so, main air masses patterns are not so easy to interpret, so we did it graphically. As you see, both in winter and summer, the main prevailing direction in Central Europe is eastward, but there are other directions such as northward in Northern Europe and from Africa. In the Balkans and Middle East, connections are ensured in whatever direction. In general, the strength of these connections becomes less intense in summer. Starting from here, we calculate complex network indices to investigate the different rules of the regions in the domain. For instance, we may be interested in finding the best sources, regions which most frequently disseminate spores to other regions, so the one associated with the high values of outstrength. As we see in purple on the left, regions in Central and Western Europe are the answer to this question. Alternatively, we may be interested in investigating the regions who receive the highest number of connections. We will see that these regions are more shifted to the center of Europe. Finally, we would like to know the regions playing as shortcuts, so the one characterized by the highest betweenness, nodes we would like to cross to rapidly connect whatever couple of regions in the domain, obtaining again a different pattern. Dealing with several centralities, we decided to rank regions according to each index, aggregate all these rankings, and identify the top ones according to such new classification. Here we show the violent plots of the distribution of rankings on the y-axis of the top 25 NUTS regions on the x-axis. What we see is that the best ranked regions, both in summer and winter, are always from Central and Western Europe, with few exceptions. In particular, if we take a look at the best three ranked regions for both summer and winter, what we highlight is Belgium, Netherlands, East France, West Germany and Austria. If we consider the last SEMRAS outbreak occurred in Europe, we can say that the results of our modeling framework are in accordance with the observations. The race TK-TTF was first observed in Germany in 2013 in early summer, and later spread to northern latitudes during the same season. Recent emergence of new virulent races of Puccinia may effectively represent a new challenge to global food security. When we started addressing this issue, we realized we needed a multidisciplinary approach, moving from atmospheric physics, plant biology, and theoretical network analysis. Whatever the method you decide to use, one take-home message from our research is that airborne pathogens of plants, such as Puccinia, need to be managed at the international scale to help designing early surveillance systems and effective control strategies. As a consequence of the last point, in the last month we decided to move from the continental to the planetary scale to investigate global patterns of Puccinia dissemination and to avoid border effects. Moreover, we started to implement more complex suitability criteria. Here, rain only was considered, but more complex interactions involving different environmental variables may be included. At the end, we would like to explore the possibility of building an epidemiological model to test the surveillance and control strategies. In the slide you can find the references, and at the end I would like to thank you for attending this presentation. For whatever question, don't hesitate to contact me, hoping to meet you the next time in Porto.